Hello, thanks for attending this presentation. I'm sorry about my voice. The cocktail yesterday with the wind that killed it. I try to go to the finish line. So thank you very much. So Atamyo is Atamyo's mission. So first this presentation will contain forward-looking statements. And Atamyo's mission is to develop, to bring to patients suffering from neuromuscular disease, with an initial focus on a family of disease called limb-girdle muscular dystrophies, next generation gene therapy. And I think that what differentiates us from other companies is that we are in the clinic. So it's the future of gene therapy, but today. So Atamyo is a spin-off from Genethon, which is a pioneer in the field of gene therapy for the last 30 years. And thanks to this pioneering work done by Genethon, we are able to tap into a very large toolbox of use for gene therapy. And thanks to the knowledge of our co-founder, Isabelle Richard, we were able to design what we call smart vectors that we believe address the key challenges of gene therapies. The first, of course, the first key challenge is efficacy, is to treat the muscular dystrophies that we are targeting, in particular limb-girdle muscular dystrophies, are very complex disease. On the left, you can see two images of patients. So it's a disease that affects the development of muscles that are used to walk, to use the arms, but also in some cases there is a specific clinical manifestation with a high respiratory impairment, or a cardiac impairment too. So you have to develop vectors that will address all these symptoms. And again, so how we address this challenge is we use different techniques. The first one that we have implemented, for example, for our lead program, is a codon optimization technology that allows to increase the efficacy by 5-fold. Another innovation that we use in other programs is the development of new capsids. So we have developed, at Genethon, Isabelle has developed capsids that, for example, are able to improve the biodistribution in skeletal muscle by several hundred folds, even for some muscle by more than 1,000 times. So all these innovation, how this translates. In our lead program, we are able to demonstrate in a LGMD R9 model, mice that has exactly the same mutation that have the patients. We're able to fully restore the function, the force of these mice with one single administration at an unprecedented low dose, which is at e to the 12 Vg per kilo. So you see on the far right up the force of one specific muscle that is fully restored. We even have a kind of doping effect with the highest dose we use. But from the mid dose, which is in the e to the 12 Vg per kilo dose range, we fully restore the force of tibialis anterior. Bottom is the escape test. You put a mice in a tube, you pinch the tail of the mice, a bit cruel, but it's able to see what's the global force of the mice. And again, with one single administration, with e to the 12 dose range, we're able to fully restore the force of the mice. So that's the first illustration how we address this first challenge about efficacy. And then, of course, the second challenge that every gene therapy is confronted is the safety profile, the safety challenges of gene therapy. So at Atamyo, we focus mainly on three organs, liver, heart, and DRG. I'm going to present how we address liver and cardiac safety. So first, liver detargeting. So we use two different techniques, innovations. We have developed a new capsid that fully detargets the liver. In this experiment, it's in NHPs comparing the biodistribution in liver AV9 versus this new capsid. But also, we are very we strongly believe that promoters also can have a very high impact on the liver safety. And for our second program, we are using a promoter called TMCK. It will be the first time that this promoter will be used in the clinic. And very interestingly, we have shown that TMCK decreases significantly the biodistribution in the liver, so helping to address or to circumvent potential liver toxicity. So the idea is indeed the objective at Atamyo that we are pursuing and accomplishing is to go from this first generation gene therapy that had muscle versus liver ratio of 1 to 3,000 to go to 1 to 0. Then the heart, cardiac toxicity. How can we address or prevent cardiac toxicity? We have developed for our third program a new promoter called ACTA. This is an image of our third program, the same vector with a classic desmin promoter and with introducing our ACTA promoter. This is a dose, single administration in the E to the 13 vg per kilo. And you can see that with classic desmin, you lead quite quickly to heart fibrosis, and just using this new promoter, you avoid such fibrosis. And for programs in which we want to fully detarget or have a higher heart detargeting, we have introduced, and it's the first time this technology is used in a clinical trial for gene therapy, a heart detargeting sequence. So this is for our program ATA100. You can see that the same vector without this heart detargeting in the blue, or in introducing this heart detargeting sequence, you can diminish radically the amount of FKRP, which is the protein, in the heart, almost to non-detectable levels. But so the idea, again, compared to first generation gene therapy that used to have a heart versus a muscle ratio of muscle versus heart of 1 to 30, to go, in some cases, we want to have a 1-to-1 ratio. This is the case for the second program, where there is cardiac myopathy, so we want to keep the expression of the protein in the heart in order to address this cardiac involvement. But in some cases, we want to fully detarget, so we go to the 1-to-0 muscle heart ratio. What's important also, this science for us is useless if you cannot produce and deliver efficiently clinical batches. So Geneton has had a 15-year really high focus on developing a reliable and efficient manufacturing process. So it's a combination of combining specific cell line with a very interesting transfection agent. And you can see that the combination of the two is on the far right. It improves dramatically the yield. So this process is licensed to Atamyo. It's also successfully implemented in our preferred CDMO, Hyposchesi, part of SK. And what's the result of that is that we're able to scale up without any batch failure this manufacturing process to our first 3 programs who have completely different serotypes, different promoters. So we can really demonstrate the platform as we have exactly the same level of quality of yield for the 3 programs. The second challenge is to reduce significantly the manufacturing cost of gene therapy. Especially if you want in the future to go to larger indications. And even for our first program, even at 200-liter scale, which is maybe not the scale we'll use for commercial batch, we are already at a COGS per patient below $100K. And we have been able to have GMP-grade clinical batches for our first 2 programs without any batch failure. So everything is possible. Of course, there is the support of the science, but because we have, I believe, a very complementary management team, really the innovation, the source of the innovation is the work done by Isabelle Richard, our co-founder and chief scientific officer. She has been a pioneer in the field for the last 30 years. So we leverage all these innovations that she has developed at Geneton. But also we have a chief medical officer or chief technical officer, experienced managers that have been up to BLA filing in the US or Europe with very innovative treatment, including AAV-based gene therapies. So thanks to the support of our parent organization, all this science, the management, so we started the company late 2020 with 5 LGMD programs. That represent a prevalence US, Europe, around 25,000 patients. Since 2 years, we've been able to start a clinical trial in LGMD R9. I can proudly announce that we will start a clinical trial for LGMD R5 this year. And then maybe I will need the help. So there is a video here, and I see that I don't know if somebody can launch the video. Yes, thank you very much. So there is no sound. So it's not fully... Yeah. So I'm sorry if there is no sound, but it's quite a good story.
Et elle m'a expliqué qu'à ce moment-là, moi aussi je me suis mis à pleurer et je me rappelle que j'ai dit que ma vie elle était fichue. Je me vois comme un guerrier mais plutôt comme quelqu'un qui... Pas un guerrier qui va au combat se battre, quelqu'un qui est un peu plus dans le combat mental.
Thank you. And so really that's what motivates it beyond the exciting science is to be able to launch these gene therapies, especially in this indication. This indication, there's nothing in development, even in preclinical stage. So we're very happy and proud to, thanks to this work, to start these clinical trials in this indication this year. So what's, to have more minutes, so this last slide, what are the key milestones that we expect for our pipeline in the next 18 months? So for the lead program, we'll complete the recruitment this year of a first dose escalation cohort, so 6 patients, 2 times 3 patients, and we'll file an IND in the US in order to open US centers this year, and we'll communicate on the first efficacy and safety data second half this year. And we'll be ready to start the pivotal part of this clinical trial early 2024. So for the second program, GMD-R5, we filed a CTA and we expect to dose the first patient second half this year and have the first safety and efficacy data next year. And for the first, LGMD-R1 is the most prevalent LGMD, representing 1/3 of all the LGMD patients. We'll file all DND, and the idea is to complete the IND-enabling studies early 24 and file the CTA IND second half 24. And of course, we want to continue to invest on the research, to continue to invest on these new technologies in partnership with Genethon, these new capsids, new promoters. Of course, this can be possible. We are well funded, well supported by Genethon, but in order to achieve all these exciting milestones, we are starting to raise 40 million Series A that will finance the runway and these programs up to early 25. So thank you very much for your attention. This is my last slide. We have 1 minute, I guess, for questions. Okay, so for the shy people, I will be outside answering questions too. Thank you very much.